Okay, we are going to start. So this is the last session of the IFCC's Scientific Division Satellite Program. It's entitled Clinical Mass Spec and Now Beyond Small Molecules, Quo Vadis. It's a real pleasure for me to introduce the first speaker, that is Dr. Anne Bendt. She's the PI, Principal Investigator and Deputy Director at SLING, which stands for the Singapore Lipidomics Incubator, and that's an internationally renowned research and development program in lipid research and technology development. And it's anchored at the National University of Singapore. So she focuses, as we could hear from her questions, on the translation of mass spectrometry-based technology into clinical applications. And she is passionate about training and education. She is also involved with the IFCC on the working group, in the working group on metabolomics, beyond many other things in which she is active. And for today, she will reveal her results on lipidomics biomarker discovery, the Ceramide case study. So Anne, the floor is yours.
Thank you, Christa, for the nice introduction. And just as a disclaimer, it's not my results on the ceramides. I'm presenting what the community has done so far on the ceramides. Yeah, so as Christa said, I'm at SLING in Singapore, the Singapore Lipidomics Incubator. And it's a research program, and everything we do is mass spectrometry for lipids and small molecules, basically lipidomics. And how many of you know of a success story where lipidomic research really has led to a translational success? There are not many, if any, but perhaps the ceramides are emerging as a success story. Let's see. So I'm going to highlight basically that and end with international efforts and harmonization. So there has been a rise in the interest of clinical lipidomics over the past years, that you see from the number of publications that are popping up, but also in more and more commercialization efforts. So there are now a number of companies offering lipidomic services. This is research use only. This is not in the clinical field. It's research use only, but at least there are companies out there who offer lipidomics as services. And also more and more research findings have been I'm lacking the word, have been commercialized and patented licensed into assays. This is primarily in the field of various malignancies, non-alcoholic fatty liver disease, of course, Alzheimer and cardiometabolic diseases. And there are also more companies now being spun off that use lipidomic readouts for to try diagnostics. And internationally, the field of clinical lipidomics researchers is organized by this Clinical Lipidomics Interest Group within the International Lipidomics Society, short CLIC. And if you scan this QR code, that gets you to our website. And this is an open invite to everyone involved or interested in this field, because it really takes a village to move this. And we need to have a dialogue with the researchers and the manufacturers and industry and regulatory and, of course, physicians and clinicians and so on. So it's an open invite to please reach out if you have an interest in clinical lipidomics. Professor Michael Vogesa and I are on the steering committee, and the other authors here as well. Now it's already quite a while ago that we published this short communication on how we see that, how the roadmap could be to really drive and translate clinical lipidomics into applications. So, of course, mass spectrometry for lipid-based diagnostics has already existed for a number of years, if not decades. This is not lipidomics, because lipidomics, as we define it, is really like all the lipids there are. But what's currently available is mass spec-based assays for lipids that are either individual lipids or small multi-marker panels, such as what we heard a lot about from Brian Cavill already on the sex steroid hormones, where really the specificity of mass spectrometry is crucial for accurate diagnostics. But also, of course, the newborn screening have been around for decades for acylcarnitines or for rare diseases such as the single lipidoses. And of course, the international cash cow of vitamin D metabolites. we recently summarized whatever is right there in clinical, in lipidomics and diagnostics in this book chapter. So this is how current diagnostics based on lipids look like. As Uta just said, obviously most of these are not measured by mass spectrometry, but by photometry or other methods. But you see basically the lipids that are currently used for diagnostics, from the lowly concentrated picomolar hormones to the more bulk lipids, triglycerides, and total cholesterol. So these are the reference ranges in human plasma. So this is the current routine. And how could this look like if we added a lipidomic readout? This would look like this. So here you see all the different lipids classes and species that we could routinely measure in human plasma, together with their rough concentrations in human plasma. And each individual line represents a different molecular species, because as you of course remember from lipid biology, a phospholipid would have two fatty acids, and depending on the saturation degree and the length of the fatty acid, you have different molecular species. So that's what's shown here. So I'm not saying that this has clinical utility at the moment. I don't think any physician would order a test like this, because what really is the actionability of this? What has happened over the past decades is that more and more people have used lipidomic readouts for cohort studies, small and large, and have in various disease cohorts to find biomarkers. And most of these have not been translated, but perhaps the ceramides are emerging as the one success story where lipidomic research has discovered something that was then further translated and might make it into routine clinical chemistry. So I'm going to use the next few minutes on highlighting the use of ceramides for cardiovascular disease risk prediction. Why CVD? Because it's the killer number one, super costly to the healthcare system. There is a traditional risk management based on cholesterol and others, but that's not perfect. And we will hear more from Christa about it, I think. And we also have okay drugs, but they're also not perfect. So lots and lots of cohort studies have happened to find new biomarkers or novel drug targets. And about 10 years ago, these researchers from Finland, from this company called Zora Biosciences, they set out to find new biomarkers for risk of heart disease, and they measured these heart attack cohorts, and they used broad lipidomic readouts, so a variety of different targeted and untargeted methods, and they landed on a group of lipids called the ceramides to have the strongest predictive differentiating power between risk of adverse events and not. And then they did the next crucial step, not just publish it, put it in a drawer and move on, but they did a validation study and really narrowed it down to these 4 distinct ceramide species, and measured these and validated these in different disease cohorts. And these ceramides, so just for the nomenclature, wait, I'll show you a structure. Okay, so ceramides. What are ceramides? Ceramides, they're circulating in the blood via lipid proteins. They're bioactive lipids, which means that in addition to their structural function, they also have a bioactive component, so they're signaling. And in human plasma, there are dozens of different ceramides. But these 4 here, so some of them have been described to be cardiotoxic and others benign, if not even protective. And here the 3 highlighted in red are the ones that are described as toxic, and the green one as the benign or protective one. And for structures, if you don't want to look at structures, just look at the Sphinx. The Sphinx is the name-giving mythical creature of the sphingolipids, of which ceramides are a part of, because they look a bit weird, and they're the most enigmatic lipids because they don't look like a typical phospholipid with a glycerol backbone. Instead, they have this so-called long-chain base, the sphingolipid base, and then attached to that via this amide bond are different fatty acids. So in the case of those 4, we see that the 3 toxic ones have 16-0, 18-0, or 24-1 attached to it, and the protective one is 24-0, which is maybe an example to show the value of mass spec, because with mass spec you can look into not just bulk of something, but really into individual molecular species and differentiate this. Also, mass spec was important for the ceramides because the concentrations are just like 1,000 of cholesterol, so you need the sensitivity of mass spec to measure these precisely. These ceramides, just after these researchers around Zura Biosciences published this, about already 10 years later, the Mayo Clinic licensed this assay, and they have it on the test menu, and they call it CERT1 for coronary event risk test. And you see the reference values, you see the concentrations are really quite low, so mass spectrometry is necessary here as a readout. And I think Uta also just mentioned that the clinician doesn't want to look at molar concentrations, but more at a score. So to enhance adaptability and of this assay, what they did is they calculate ratios of the benign to of the toxic to the benign ones, and take the population quartiles. And according to where you sit on the quartiles, there's a point system assigned, and that's how there's a risk score assigned on a 1 to 12. Did I explain that somehow coherently? Okay. So the name CERT1 implies that this was not the end of the story yet, and yes, indeed, there's already a new one called CERT2. So the same researchers with a few additional ones improved this assay already by adding 3 additional lipid species, phosphatidylcholine, which are the most abundant phospholipids in human plasma. And this new test is now called CERT2, and it's been shown to outperform the CERT1 in these large epidemiological studies. So the hope is that this can really lead to more personalized risk prediction, basically identifying patients at higher risk so that they could qualify for stronger treatment, for example, with the PCSK9 inhibitors and so on. So also this assay then got licensed by by Quest. It's not rolled out yet, but they have the license and it's ready to be used. But I think everyone is waiting for the reimbursement code to materialize. And so this is where it stands regarding to a commercialization. The test is easy. We had a brief chat on this over breakfast. So you just need 10 microliter of plasma, which is nice, and you just shake it with 500 microliter of solvent. And the deuterated internal standards exist commercially. So for these 4 ceramide species, the 4 authentic internal standards can be added straight away to the solvent. A bit of centrifugation, and the supernatants can directly be injected into the LCMS, and then you can run external calibration curves. And here in this table, you see how they assign the score by the point system, depending on where you stand on the quartiles. 100%. And so that's how it works. It's pretty fast. I think the run is about 7 minutes or 10 minutes of the LCMS. So this is all LCMS by multiple reaction monitoring, all targeted. So this test then has been so there are many studies now comparing the different ceramide tests. I'm just showing you one study here on the left side, which is based on 999 Austrian people, where they compare the performance of the new ceramide test against the traditional risk management. So in black, you have the basic score, age, gender, smoking, blood pressure, and total cholesterol. And then if you add the ceramide, the CERT2 score, that's the red line, or even a combination of both, it really outperforms the traditional risk management to identify those patients at high or very high risk of cardiovascular events and death. So, this has now been validated in even more studies and prospective cohorts. It looks like the improved SIR2 really outperforms the conventional diagnostics, and it has been tested on about close to 30,000 people from various with various comorbidities. So young, old, obese, CVD, yes, no, diabetes, renal failure, and the elderly. And so my question earlier today: at what stage would a clinical lab now really take this on, and what else is the evidence that needs to be provided to make a decision if it's useful to roll out this test? I don't know, but so the Mayo Clinic has it, and Jeff Moosen, who is in charge of running this assay at the Mayo Clinic, he wrote this editorial, and so sort of saying lipidomic. It's not lipidomics, but it's like so has made it into routine diagnostics. So we would not call this lipidomics, and we discuss this all the time. How many things do you need to measure to be omic? In this case, it's not a lipidomic test because it's not hundreds of lipids, but it came out of a lipidomic research and got translated into the small panel of a few lipids. Yeah. So this led to a lot of. Let me walk you through this first. So advantages and disadvantages of this test. So what's really nice about this test is you only need 10 microliter, but what's even better is the ceramides seem to be really stable, and that does not hold true for many of the other lipids. So for oxylipids, this would not be possible. And for most lipids, having the blood stand on the bench for extended periods of times would not be that good, but the ceramides don't seem to care that much. And 8 hours at room temperature on the bench is sort of okay. And the sample prep is easy. I've showed you how easy it is. Can easily do it automated in 96 well plates. And yes, they are commercially available, deuterated internal standards. Peak integration is easy, of course, and mass spec becomes more and more accessible. But what's needed to demonstrate value? So some people here say that what would really convince them is an intervention study, like a randomized control trial, where you stratify the people into some of the patients get treated according to the traditional risk management, and the other arm gets treated according to the ceramides, and then you check for improved outcomes. It's of course the hardest, and I don't know who would now go about doing this and pay for this, but what's also still lacking is really reference intervals and a more thorough understanding of the biological variation of these analytes in populations, especially in diverse populations, because the reference intervals on the Mayo Clinic isn't based on Americans, but we haven't really derived reference intervals in other populations. And in the discussion just now, also someone said physicians would only order something once it's in the guidelines. So obviously the ceramides are not in any guideline yet. And also what really is the actionability. So there's no specific treatment that specifically just lowers the ceramides. They are modifiable by Mediterranean diet and exercise, but what isn't modifiable by Mediterranean diet and exercise? And so how actionable is it then really? But at least it's actionable in the way that a patient who hears that they have really high levels of ceramides, they might be more prone to listening to suggested lifestyle choices. Yeah, and then ensuring accuracy across time and space, right? I mean, what do we do about harmonization, standardization? There's obviously no proficiency testing or external quality assurance system yet. Would the industry ever step in and make a kit based on what? On CERT 1, on CERT 2, or wait for CERT 3? Or when would anyone step in and commercialize this further? So yeah, it's easy for us in the lab, but in the research lab, but it's not plug and play yet. So anyway, the community also really, the research community is very interested in measuring the ceramides as well, and that was started actually by Marcus here in the audience about, I forgot when, maybe 5 years ago or something, somewhere there. Feels like 20 years ago, but it was a a large community effort of learning from the our clinical colleagues who run ring trials and interlab studies all the time. And as researchers, we typically don't do this, but the community came together, and the lipidomics community is one that's quite interested actually in harmonization and standardization, more so maybe than some other communities. So the International Lipidomics Society, together with Lipid Maps and then NIST and Avani and Cayman, came together to organize these ring trials. Here you see I don't know, we call this color beige, everyone who participated in the ceramide ring trial. And there's also another one on vial acids. And you see it's about 35 different labs all over the world. And so there was the ceramide ring trial. It looks like this. So I took this slide from Dr. Federico Torta from Sling, who together with Marcus is the lead driver of this effort. So we call this phase 1 because this is the technical phase of the ring trial, and it was all about just measuring reference materials and measure absolute concentrations of these 4 ceramides in commercially available reference materials. So everyone was asked to measure these 4 ceramides from the CERT1 and the corresponding 4 deuterated internal standards. These are exactly those 4 that are CERT1 on the Mayo Clinic test menu, and the samples, the reference materials that were measured were the NIS 9050 and then also the new plasma suite, which is the high triglycerides, the diabetic, and the young African American to account for potential matrix effects and ethnic components. And what was received by the participants is everyone got these 4 reference materials and also 1 vial with the premix internal standards and the non-labeled standards for the calibration curves, and 1 SOP. And the SOP, as you all know, an SOP is never as good as you think it is, and it went through multiple rounds of rewriting it and getting feedback, and in the end it was a pretty good SOP, I think, and participants were free to use that SOP or their own. And then everyone reported their data in an Excel sheet. And the paper is now out, so if you scan this QR code, it was just published about a month ago, I think, in Nature Communications. Here I just show you 1 figure, and that is the figure of all the different, like the concentrations for those 4 different ceramides as reported by the 34 different labs, and in NIS 9050. And you see in red everyone who followed the SOP that was developed by Federico and team, and or their own method as others. And great news, I think, is it looks pretty similar. So it doesn't really matter if you use that SOP or your own, if you're a lab who's good in measuring these things and have good protocols yourself. So the intra-lab variation here was about 4% CV, and inter-lab was about 14, which is great. So that just shows that everyone can do their own thing if they use shared reference materials and the same internal standards. So that's basically the take-home message for this particular technical phase of the ceramide ring trial. Christa, how am I doing for time? Okay. Is it a thumbs up or 3 minutes? Okay. So now with this, I'm giving you a short overview of where we stand internationally with ring trials. So the ceramides, that was just concluded phase 1, which is measuring the absolute concentrations in those reference materials. And now there's an appetite to move into phase 2, which would be really to define the reference values in plasma of the free-range humans, all these populations out there, and measure worldwide to really come towards reference intervals. That's now hopefully slowly starting. And then the bile acids, this other ring trial I briefly mentioned is about to be launched. This is 18 compounds. Cayman stepped up as a partner, and then within the International Lipidomics, Clinical Lipidomics Interest Group, we also thought we do one that is more towards lipidomics. So we designed one Because it's somewhat easy to do 4 analytes, and 18 analytes is more complicated, but none of this is the lipidome yet. So we designed one that's more descriptive of the lipidome, and that is 160 lipids from 11 classes, and that tremendously complicates basically everything. It took a long time to design and launch it and to agree on methods. In this case, four different methods are allowed, 4 different workflows are allowed. And we designed and launched it, and currently data analysis is happening, and it's I don't know how many million data points. It's gonna take a while to come to any conclusions here. Yeah, and everyone measured exactly the same materials as I just mentioned, plus also 1 extract just to account for, just to take away the potential error of extraction. So this brings me to my summary. I hope I could convince you that there's a vast diagnostic potential of clinical lipidomics. Some lipids are not only associative, but probably even causative for diseases. The current applications still are firmly anchored in the R&D or research use only space, discovering biomarkers and understanding pathophysiologies better. But potential, the ceramides, I mean, the jury's out. Do you consider the ceramides already success stories? So this is, and yeah, I highlighted the community-led trials, and it's of course way more complicated. And I invite you to read this little article Professor Vogeser and I wrote to highlight all the other things that are even more complicated, which I didn't have time to touch on, which many of you are of course more aware than us researchers are. So what really are the challenges translating an RD method into a diagnostic test that could really be adopted? And how do we ensure quality across time and space, especially for moving from single lipids to small panels to larger panels? And how do we deal with QA and QC over time and space? And the lack of reference materials, of course, internal standards and harmonization and so on. Yeah, this is where we stand with this. And with this, this is how Sling currently looks and our funding. And if you are tired of the weather in February in Europe, then please come to Singapore last week of February for our Lipid Symposium, which this round for the first time is co-organized with MSACL, and Amber Harold from MSACL is here, and I'm extremely happy that we managed to lift off a 1-week program with 2 days of instructional workshops around clinical mass spec, followed by our normal Lipid Symposium. Thank you very much.
Thank you very much. There is time for one question. Yes, please.
So, hi. Thanks very much for the nice talk. I have 2 questions actually quickly. The first one is, the ceramides have a great potential to be implemented in the clinic, right? I read somewhere this week that basically HDL and LDL routine methods are not as informative as ceramides. Do you think at some point ceramides will beat the HDL and LDL parameters in the lab? And the second question is more technical. So for the ring trial in lipidomics, you are trying to analyze now hundreds. How do you harmonize the software? Because everyone will use their own software and stuff, and it's more tricky. Thank you.
Yeah, thank you. I answer the second part first. So the software, so the data analysis for the lipidome ring trial happens centrally. There's this one poor soul who does the entire data analysis. And everyone, and there was a spreadsheet where everyone had to just punch in basically peak areas, and then it gets auto-calculated. By the way, for that trial, obviously we did not have 160 authentic internal standards, right? That's one problem with that lipidome, because we don't have 160 internal standards. We only had 35 or so. So 2 to 3, sometimes 4 per class. Yeah. And the first question was if I think that ceramides would replace cholesterol. I leave that to Christa. I think initially it's a big task already to just have an add-on, and we're far away from replacing something. And I think the one figure I showed from this Austrian cohort the performance is best if you add on the ceramide on top of the traditional risk management and not just go like change everything and don't do cholesterol any longer. Yeah. Did that answer your question? Yeah, thank you.
So thank you very much, Anne, for this very interesting talk here.
Okay.
It's also a big pleasure for me to introduce Claudia Swart. Claudia is not live present. She has a virtual presentation, so we see her looming. Claudia, you can hear us? She's muted. Okay, Claudia, can you unmute yourself?
Can you hear me now?
Okay, yes, yeah, we can hear you. Okay, so Claudia, she is working at PTB, that's the National Metrology Institute in Germany, and she is also the PI of what was named Cardiomet. That was a huge consortium. It was an EU subsidized program working on an infrastructure for cardiac markers. And today she will present towards an MS-based reference measurement system for cardiac troponin. So it's up to you, Claudia. I hope you can upload your presentation. And please take the floor.
Okay. Thank you, Christa. Can you see my presentation?
Yes, we can.
Thank you. So I'm sorry that I can't be with you today, and I hope you can understand me because I caught a very bad cold during the last business trip. But thank you so much that you give me the opportunity to tell a little bit about our work on the Cardiomet project and the results that we had for MS-based reference measurement procedure for cardiac troponin. So the development was mainly done by my colleague Danya Kufus here at PTB, and my colleagues Maltem Asicioglu and Merve Öztürk at TÜBİTAK UME in Turkey, who did the LC-MS method. So first of all, as many of you might know, troponin is a protein that's located in the heart muscle, and it is released when the heart muscle tissue is damaged. The most prominent disease here is the myocardial infarction, for which this is one of the main biomarkers. Troponin itself consists of 3 different subunits. Troponin C, it is not specific for the heart muscle, but can also be found in skeletal muscles, while troponin T and I are specific and are used as biomarkers for the damage of the heart muscle. Why is harmonization or standardization necessary for this biomarker? When you have a look here at the PT scheme from the reference Laboratory for Bioanalytics here in Germany, RFB, which is one of our reference laboratories in clinical chemistry, you can see that of 432 participants who received 2 different samples, the spread of the results is quite high. So how this is done, every participant gets sample A and sample B. Sample A usually has a concentration that is close to the clinical cutoff, and sample B is an elevated one. And you can see here that we have a coefficient of variation over all results of about 60%, which is quite high. I must also add that for troponin, as is the case for many other biomarkers, the main important part is not the absolute concentration, but the change over time. So usually for the myocardial infarction, you have a regime taking one sample at 0.0 and after 1 or after 2 hours. And then you see how this is increasing or decreasing to see if you still have an active myocardial event. So now it has started to become that point of care tests came to the market. So from now on, it is possible that the first test might not be taken in the clinics, but somewhere else. Currently, this is not possible because the results from one assay is hardly comparable to the results of another assay, as you can see here. So it's very important to have at least harmonization, but ideally standardization of the results of the different kits. So how can standardization be achieved? So that the optimal version is to have a traceability to the International System of Units, DSI, through different steps. So usually you have your routine methods that are measured in the clinics every day, hundreds, thousands of them. The results here are quantified using a calibrator, which ideally got a value assigned using a value transfer procedure by the manufacturers from a secondary certified reference material to the calibrators. And this certified reference material is value assigned using a secondary reference measurement procedure. And ideally, we have also a primary reference material. This is usually or the purified protein or whatever, which is value assigned using a primary reference measurement procedure, which then links the result for the primary reference material to DSI. For protein or in the clinics, it's the main important units are mole or kilogram, while mole is preferable because at least for proteins, it's quite difficult to define a molar mass and have a defined mass concentration. So what is a primary reference measurement procedure? I will cite now from the minutes from the 5th meeting of the CCQM, which is the committee at the Meta Convention, which is responsible for chemical and biological measurements. And they have two different steps. So one, and the most difficult one, I think, in the clinics is the definition of the measurand. Most strictly spoken, this is not part of the reference measurement procedure itself, but that's the basis for everything. So in case of a protein, we have the amino acid sequence, which is often known. And for many proteins, this is also quite stable, give and take 1 or 2 amino acids that were changed due to some genetics variations. What we often see then in real biological matrices is that we have degradation, we have post-translational modifications, such as, for example, glycosylation, which also changes the function of the protein. So this is definitely part of the measurand that we have to take into account. And then we have also complexation. In case of troponin, we see that's a trimer. We have 3 different subunits. When this is released in blood, it starts to degrade. So often it's only a dimer that you can see. So it's the complexation of troponin I with something, mainly C. And we have often a real mixture of molecules. So as I said before, we have no defined molecular mass. The next step is then for a reference measurement procedure that the whole operation can be completely described and is completely understood. So you have no step, but you don't know what's happening. And every step, of course, has an uncertainty component, which has to be known and has to be taken into account to have a complete uncertainty budget in SI units. That's also a little bit different to many routine methods that you don't have a complete uncertainty budget, but it's often more or less reproducibility or repeatability, and all the other steps are not considered when you have the coefficient of variation. So what is often used in chemistry or biology is isotope dilution mass spectrometry, which I will go more into detail in the next slides. So what is for troponin? So we have our plasma or serum sample. This here should be our analyte, the troponin, and the concentrations are really low. You have seen in the PT schemes, we are in the nanogram per liter range. So that is too low for mass spectrometry to detect directly in such a complex matrix. So it's necessary to enrich it. For this, we used magnetic nanobeads that had either acid residue, or for the other thing, we have protein G, where we have captured or bound the antibody to the nanobeads that is specific for troponin I in this case, so that it can be captured and enriched. So as I said, we do isotope dilution. That means that we add the analyte in this case troponin I, with a different isotopic composition. In case of LC-MS-MS, the C12 that's usually inherent in troponin is replaced by C13. And this internal standard is ideally added before you do anything with your sample, so that the internal standard accounts really for every single step in your sample preparation, separation and detection. So it's added here at this step so that both are enriched together, the natural one and the internal standard. Then it's difficult to remove it from the beads again. So the troponin is digested. In this case, it was used trypsin. And then specific peptides are selected that are stable and specific here for troponin. And in this case, there were two different peptides you can see here. And these were then used for quantification of the troponin. In the project, our colleagues from UME also tested more a cheaper version of isotope dilution, because those internal standard or complete protein isotopically labeled is quite expensive and not always available. So another approach is to first enrich the analyte from your sample, do the digestion, evaporation, the same as before, and then you add the peptides in C13 labeled as internal standard. Of course, you can see that now we have this whole part in the sample preparation that is not accounted for by the internal standard, but still it accounts for everything that occurs in the citation source or in DMS. And again, the two specific peptides were detected and used for quantification. Within the project, we also used a completely different approach to this one. We used inactively coupled plasma mass spectrometry as the detector instead of organic mass spectrometer. ICP-MS detects only elements, so because of the high temperature in the plasma. Sorry. Everything is burned down to elements. But that means also that we don't see much from troponin in the end, because there is only carbon in it and nitrogen and oxygen, but that's nothing that you can easily detect in an ICP. So what do we do in this case? We add another internal standard, and this internal standard is labeled with a lanthanide. So you can see here, this is the label that we produced, and you can add here lanthanide isotopes as you like. And via this group, this label binds to the cysteine residues in the protein, and then you have also an internal standard. Why did we choose lanthanide? Because ICP-MS is very, very sensitive for lanthanides, and this is an element that you usually don't find in biological matrices, so there won't be any background or interferences from this part. So again, this is enriched together with the analyte, and then we still have the problem that we wouldn't see the biomarker in the original sample. So we have to add another label, a lanthanide label, a different isotope in this case, from the same lanthanide that we have here for the internal standard. And then, yeah, we have both on the magnetic bead, which is separated, enriched. Again, we used enzymatic digestion and detected us with the ICP-MS. So you can see here we have again our LC for the separation of the peptides, and then ICP-MS for the detection of the lanthanides. Of course, when you do digestion, you have to check the digestion kinetics, if it's complete or if you have degradation of your peptides and so on. So for the digestion of troponin I with trypsin, the colleagues at UME, they used different buffers in which they did the digestion, different ratios of analyte to the enzymes, so 1 to 100, 1 to 50, 1 to 25, and 1 to 10. And you can see here for 1 to 25 and 1 to 10, the results is best. That can be because you have a lower background if you have not so much enzyme here, and it's only necessary to add this part. You always get self-digestion also from the trypsin, so it's good not to add too much. And then you have to check the time so that you ensure really complete digestion. You can see here for the overnight digestion, the results were the best. What I also tested then is to see if they should add the trypsin in one step, or if it's better to add it in 2 steps. So here, this is for 1 step, this is for 2 steps. You see the overall release is not really different, but the uncertainty is a little bit different. For the ICP-MS method, we had to use a different digestion method because with trypsin, we get only one peptide that contains cysteine due to the points where it cuts. When we use Glu-C instead, we get 2 peptides, which is preferable so that you can verify the results from one peptide with the results from the other peptide. And you can see here the 2 different peptides that we were looking at. For one peptide, we get more or less a plateau at between 30 and 40 hours. For the other one, there was no complete release, and we could also see that it started to degrade after a while. So in the end, we still ended up with one peptide, but nevertheless, it was a try. So then we did the method validation for all 3 of the methods. We checked if the calibration curve is linear. We checked the accuracy, the precision, potential carryover in the liquid chromatography method. Also the recovery, this is especially important with the magnetic enrichment. And of course, limit of detection and quantification. For this, different QC samples were prepared. We used troponin-free serum that we procased from HiTest and added the pure troponin complex from NIST. So this is a primary reference material that contains the CTN complex isolated from human heart. Unfortunately, this material is currently not available anymore, so we had to use what we had left. But NIST is currently working on that, and at the beginning of next year or during next year, the follow-up material will be available. So different concentrations were prepared, and you can see here that we, despite the enrichment, we do not reach the clinical cutoff value concentration. We are now in the microgram per liter range, in the low microgram per liter range. This is still clinical relevant for a clear myocardial infarction, but it's far from what the immunoassays can do and what is necessary for a quick diagnosis. But this method is never meant to be used in clinic, so it's might be not that problematic, but that's something we still have to work on. So if we have a look at the different ways of calibrating this LC-MS method, we have one calibration using the whole troponin labeled as internal standard, and then the peptide calibration. Regarding the linear range, they are quite similar. The repeatability is similar. The precision is somewhat less with the peptide calibration, but what's the real difference is that the recovery for peptide calibration is really low. And that is because you add, if you lose something in the whole step of enrichment, digestion, and so on, you cannot account for that with the internal standard, but this is added later on. So in this case, you can see that peptide calibration is not a very good choice. They did a workaround because this recovery seems to be quite stable, so they used a correction factor. But this is also very high, and with a correction factor, you introduce another uncertainty component. So for the very low concentration peptides where you need enrichment and so on, peptide calibration has to be regarded as far inferior to using the whole protein as internal standard. For the ICP-MS, we were really surprised when we saw the results because you can measure in the low nanogram per liter range and below lanthanides. You can still measure in the nanogram per liter range if you use the labeled peptides, pure ones. If you have a look at the labeled troponin or the peptides released from the labeled troponin, the limit of quantification decreases dramatically. So it seems that we might have some problem with the digestion using Glu-C. and it's still less in serum, which is maybe due to some matrix effects from the proteins from the serum. So we still have to work on that and find the reason why it's so difficult really to release the peptides and really detect the peptides in clinical samples. Yeah, as I said, reference measurement procedure includes a complete uncertainty budget. So just an idea what can all be part of such an uncertainty budget. Of course, you have your sample. You have the homogeneity of your analyte in the sample. You have the stability of your analyte. That is also often a problem. And then, so as we do everything with traceable to DSI, we do usually no pipetting. We do everything by weighing. So you add uncertainty by weighing, but it's far less than what you get from pipetting. The same is true for the calibrator. So you have the weighing of the calibrator. Usually you have an uncertainty that's associated with the value assignment of the calibrator. In our case, then the digestion is the next step. So you have the reproducibility from the digestion. It has to be complete or at least identical for your internal standard and your analyte, which can be a problem. For example, if you have a highly glycosylated analyte and you use a recombinant protein as internal standard, which lacks this glycosylation, it might behave completely different during digestion. And your released peptides have also be stable. Otherwise, you will never get a stable signal in the end. And then, of course, the measurement itself adds also to the uncertainty. You have systematic errors, you have random errors. The separation might not be complete. The integration is also adds also to the uncertainty. So when we compare all those methods we had a look at, the LC-MS method used by UME was based on a method published by NIST in 2018, but they used nano-LC and improved also the method a little bit. So the limit of quantification is about half that was what was observed by NIST. It's not far better, but still every step is good in this concentration range. It's comparable with the peptide-based calibration. It's far less good with the ICP-MS. So as I said, we still have to find the influence that the matrix is having here and why. Then we have the recovery, which was only 30% for the NIST method, which could be quite improved by our colleagues from UME. But as the recovery here named for the peptides is using the correction factor that I talked about before. And for the ICP-MS, we have also recovery of over 90%. So this is quite good. The uncertainty is comparable for all the LC-MS method and was only half for the ICP because the ICP itself has quite good reproducibility here. So as I know that many methods are out there that intends to standardize also using peptides, and we had quite good experience with other proteins in the past. We have currently had a ring trial at CCQM using hemoglobin, and you couldn't see any difference between using the whole protein as internal standard and the peptides. But as we saw here for troponin, that was quite different. So what's the advantages when you use the whole protein? It behaves very similar to the inherent analyte. It accounts for all sample preparation steps except the sampling because, yeah, it's not in the blood, but everything from them on, it's accounted for. And it also accounts for changes that might occur during detection. So you have different fragmentations and so on in the MS. The disadvantage is that the internal standard is very expensive. So we talk about tens of thousands of euro for just a milligram. And depending on the production system, it is potentially not identical to the analyte. So you can use hex cells, for example, then you get a glycosylation, but even the phosphorylation, so for the troponin, it's more the phosphorylation that might be lacking. If you use E. coli, you usually have nearly no PTMs. So that's a little bit dependent on the production system. For the peptides, it's comparably cheap. So it can also be afforded by more routine laboratories with more samples than we do at the National Metrology Institutes. It's often more stable in solution than the whole protein. It is less influenced by post-translational modifications because usually you use signature peptides that do not contain any PTMs. And it accounts also for changes during detection, as do the protein. What it does not account for are the changes or losses during sample preparation, such as extraction, enrichment, and digestion. So to conclude my talk, the use of labeled peptides as internal standard is a low-cost alternative to labeled intact proteins where feasible. Not for all proteins this is feasible. However, especially for low abundance proteins, it does not account for the extraction and enrichment, and this might be a really large contribution to the uncertainty. So that's preferable here to use the whole protein. We used LC-MS-MS and LC-ICP-MS as complementary approaches, and this allows for mutual verification of results. That is something we still intend to do as soon as we have figured out the limit of quantification for the ICP-MS method. A reference method measurement procedure with an LEQ at the 99th percentile, which is the clinical cutoff, is unfortunately still lacking, but we haven't given up. We're still working on that. And we still have one step in here that we have to investigate in more detail. This is the immunoenrichment, because we still have to see if all clinical relevant forms are really extracted that way, or if if we get changes in the composition of the mixture by this immunoenrichment step, as not all variants of troponin, degradation forms, etc., pp might be enriched the same way. Yeah, last but not least, I want to thank my working group at PTB for the work, and the bioanalysis laboratory at Tubitak Ume for their contribution. The members of the IFCC working group, we are currently working hard on establishing at least a secondary reference material to allow for harmonization. So there's a lot of work going on. And I also want to acknowledge the European Union's Horizon 2020 program and Euramed, who enabled us in the CardioMed project to do all this work. And I want to thank you for your attention, and I'm happy to take any questions.
Thank you so much, Claudia. Are there any questions? Claudia, can you, because the clinically relevant range is even lower, of course, you mentioned it briefly. What are the next steps in order to reach the goals? Because especially for non-STEMI, we need those lower limits. Is there a strategy?
Yeah, as I said, we have currently, we are working on the secondary reference material that can be used also with the immunoassays. This is a plasma material produced by NIST. And this material has 4 different levels. So from the lowest level, it's about the cutoff values up to very high levels for active myocardial infarction. And these were produced by diluting from the high concentration pool with plasma from healthy people. So if we assign the higher concentration, it might be that we can value transfer the SI traceable value from the higher concentration also to the clinical relevant concentration. So that's the idea we currently follow, as I don't see that we can reach 5 nanogram or 10 nanogram per liter with MS currently.
Okay, thank you. Other questions? If not, Claudia, we wish you better health. Take care of yourself, and many thanks for joining us here. Thank you very much.
Thank you for inviting me.
Okay, I have a final lecture on demonstrating, on demonstrating the clinical value of a multiplexed apolipoprotein panel. And we did that in an already published randomized control trial, the Odyssey outcome trial. These are my disclosures. Of course, the study was set up by the pharma industry quite some years ago, and we retrospectively measured the apolipoprotein panel in this study. And I want to give you some background. Why would we like to do this? And reflect on the clinical performance and the clinical effectiveness. And yeah, the reason why we wanted to do that is because looking into cardiovascular risk management, as it was told by Anna also, it's still the #1 killing chronic disease, even at the global level now. And if we look into how successful are those pharma trials, we can see that there is a lot of remaining residual cardiovascular risk, notwithstanding the fact that potent statins, for instance, and also the newer medications on which I will reflect, are allowing them to get at the on-treatment LDL cholesterol level, because there is a rather blind focus on only LDL cholesterol. And they do approach that, but nevertheless, patients keep getting infarctions and keep getting adverse events. So more has to be discovered and a more holistic approach is needed. And here you can see on the right-hand side a clinical care pathway. So what is the target population? What do we biochemically normally do? Mostly still a lipid profile only. And of course, in the context also of other clinical and imaging variables, clinicians make a decision. And it's only in case of a high LDL cholesterol and maybe a risk score, they embed also other risk factors in a scoring system, that they give rather generic treatments to the patients. And statins are still the #1. Of course, they are still looking for better medication in order to address that residual risk. And then we know currently, as because of time I cannot include these slides, but the residual risk is still offered. It's about 70% still. So that means that we are doing things in a suboptimal way, and we should address it. We should also have attention for the fact that we often include not enough women. They have been neglected, whereas women have another constitution, and also remnant diseases, for instance, are not taken into account. So therefore, we want to do more. And as you can see here on the right-hand side, if our idea was we should maybe look into a comprehensive apolipoprotein panel because of that residual risk, and then taking into account what you normally do for proper test evaluation. So if there is an unmet need, the residual risk which is still there, then we should go through that whole circle. 5 key elements have to be addressed. If you think you have a better marker, ceramides, or in this case, the apolipoprotein panel, we should go through all of them. And of course, it's important for getting reimbursement and acceptance, and also, of course, acceptance in the guidelines, that clinical performance and clinical effectiveness will be demonstrated. And that is what we will try to explain, how we did that and which study approach that we took. And here only in this infographic, I want to show, it's a very rudimentary one, apolipoproteins. These are the functional proteins on the lipoprotein particles. And they all have a clear role. And as you can see here in the left corner, they are either modulators of LDL particle clearance or of the clearance of triglyceride-rich remnants or lipoproteins. And A1, for instance, has a role in the reverse cholesterol uptake. And here you see the cascade. Liver cells, they synthesize ApoB. It's packed with triglycerides in triglyceride-rich particles, which are secreted. And then we have that VLDL, ideal LDL cascade, in which we get rid of the triglycerides by lipoprotein lipase, for instance. And this is then activated by, for instance, ApoC2. It's inhibited by ApoC3. So all these apolipoproteins have a clearly understandable role in that lipid metabolism. And you should also understand they jump from one particle to the other. So it's not you can't determine them, in fact, with a snapshot. So having a more complete picture of what they do, because they are so interrelated, made us decide that we should work on a profile, a complete profile, and consider that profile as a multi-marker. It's all in one package. And on top of, I briefly touched upon A1 and B, who is in that VLDL, IDL, LDL cascade, and then the C2, C3, important activators or inhibitors of lipoprotein lipase. There is also an independent, atherogenic, and genetically determined apolipoprotein, and that's the apo-a, which you here see again, with it's an LDL-like particle, LPA, with that typical apolipoprotein small a, with its kringle size polymorphism. And it's now clearly demonstrated and also picked up by the different professional societies that we should measure it. In Europe, they say once in a lifetime at least, and early on, already in youth, because of its well-known association with cardiovascular disease. And you cannot predict its level based on the current lipid levels. And here you see again the diagram. There is a real high risk. If you neglect it, then you will underestimate the cardiovascular risk. So it's already advocated to measure it, whereas the new drugs, either small interfering RNAs or antisense oligonucleotides, these will become available from the quarter of next year, the first quarter, of next year. So the results will be actionable, and so therefore there is already a lot of attention to get this done. So therefore we composed a panel encompassing 9 apolipoproteins and also the ApoE phenotype. This is a polymorphism. So, and we wanted it to go into these steps: clinical performance, analytical performance. We have already published, and I cannot repeat that, the analytical performance that has been conducted. So we wanted to look now into what is the ability of this panel, this multi-marker panel, to indeed predict a particular clinical condition. So the residual cardiovascular risk, which we want to address. And we also wanted to know what is then the effect on health outcome. So really the endpoints, which is not an easy task. And so we want to address this, and therefore we have been searching quite a lot to find clinicians, cardiologists in this case, which were interested in exploring this with us. So after a big search, it was an odyssey an sich, we came out that the cardiologists, and it's an international steering committee still, from the Odyssey outcome trial, which has been published in 2018 in the New England Journal of Medicine, was prepared to work with us. And we wanted to do retrospectively measuring it, whereas that trial has been published, as you can see here. So the population was, and the goal was first, they wanted to test PCSK9 inhibitor, alirocumab, which would further reduce the LDL cholesterol. So that was their focus, and that was published. The population that they had was mainly a hypercholesterolic patient population, so you can see the characteristics here. They all had an acute coronary syndrome, at least more than 1 month since the event. And these are the lipids. So as you can see, they are quite low because all of them were already treated with statins. So this study was meant to further reduce LDL with alirocumab. And they also had an equivalent of ApoB, which they wanted to address. It's important to consider that all these patients, and the study that I will show, these were already optimally treated with normal statins, as you can see here, or treated until what they could tolerate, because not all of us can tolerate statins in an easy way. The follow-up median was about 2.8 years. And the composite endpoint, so we talk about survival now, is death from coronary heart disease, non-fatal myocardial infarction, fatal or non-fatal ischemic stroke, and unstable angina requiring hospitalization. And what they could reach with this study, as you can see here on the right-hand side, if you look into the absolute risk reduction, it's in fact not so much. And you can see there is still remaining a residual risk, which I already told you. So we were hoping with the apolipoprotein panel that we could contribute further to understanding what should be targeted in the future, because they did not target specifically APOL small a or lipoprotein small a. I must say alirocumab does have effect on it, but it was not specifically meant. It was majorly driven by also LDL cholesterol lowering this trial. And all of these patients, as I said, all what is included, they had already statins. And we had to look in the added value of an apolipoprotein panel on top of what they, or this highly selected and highly treated patient population. So here you can see an infographic of how we set up our method. This is a semi-automated variant of the reference measurement procedure, which I talked briefly upon this morning. And yeah, you can see we work with a Bravo type ELISA plate. We are plating the samples in ELISA plates. And as you can see, we take the normal route of addition of silk peptides, denaturation, reduction, alkylation, digestion, cleanup with solid phase extraction, LC-MS measurement, and then mass quantification. Okay, so having said that, it's today about the clinical questions. How can we prove that this panel, this ID, is a good ID? So we wanted to look into clinical performance, and as already was mentioned, we do realize that we cannot get rid of the lipid panel. So we immediately positioned it as we should look into its added value on top of serum lipids. And how does it, how predictive is it for major events and cardiovascular death on one hand, and clinical effectiveness means really you look into then the end terms. And in this case, because it was a pharma trial, we looked into, can apolipoproteins help us predicting who should have benefit? So can it help to predict treatment benefit with alirocumab? Because that were the two arms, either with or without, in order to further reduce cardiovascular disease risk. Here you can see, and I will immediately show the numbers, we measured about 6,000 patients in the placebo group and 6,000 in the alirocumab group. And here you can see some frequency distributions. So as you can see, men and women, women are only 1/4, and 3/4 of men were included. And for APOL small a, you have the typical distribution skewed to the right. We also looked into the others. APOL B, it's peculiar that you see they are quite low compared to the general population. Normally, if you don't take statins in the general population, the median is already 1 gram per liter. So you can see it's a highly treated population. The ApoB is indeed low. And so this is for C1, the other apolipoproteins, the distribution. So most of them are rather normal. Okay, let's now focus on the first question. What is the clinical performance of this apolipoprotein profile panel? And how does it give an idea of the events in the future? And as mentioned, the endpoints are here. And we had to do this. I told about 6,000 individuals. And we looked first into the placebo group, which was not treated with alirocumab, in order to predict the clinical performance. And in that group, you could see that we have about 12.2% first MACE, and we had 2.6% cardiovascular death. And what we had to do, and of course we needed statistical advice from experienced statisticians, and we used logistic regression analysis with natural cubic splines to estimate the continuous relationship between these profiles, because we wanted to look apolipoprotein profile compared to lipid, and then also as an add-on, and in relation to cardiovascular outcomes. And this is what you can see. The lipid profile gives an area under the curve of 0.58, and we can improve, because it's again a very generic approach, considering all the apolipoproteins. We have about 0.65. Of course, it's still not ideal, but it's not overlapping, so it's significantly better. And if we do the combination, you can see that's more or less not further improving. But it means that apolipoprotein as a profile, it further improves the area under the curve of the lipid panel in this very selected, highly treated acute coronary syndrome study population. So, okay, we were glad already with this finding. And then if you look into the individual apolipoproteins, because that makes it rational why we chose for a panel, then you can see that, in fact, no discrimination can be made. So individually, they don't add a lot, not even the apo-a. And if we look into predicting the second endpoint, cardiovascular death, in which we had much less patients, then you can see with the lipid profile, this is the area under the curve, and the apolipoprotein profile is significantly improving compared to what we have for lipids alone. And if you do the combination, it's even further improving. So here we can say, if you look into clinical performance, that this apolipoprotein profile improves further the area under the curve in this, compared to the lipid panel, in this very selected acute coronary syndrome category of patients. And here you can again see the individual ones. Now in the relation to cardiovascular death, that this is hardly discriminating and not contributing in the way as we saw with the panel. So regarding clinical performance, we can say that we see indeed, and we hope that this will be convincing enough, but it's still on average that you see this effect, whereas we also wanted to see what is now the effect on outcome. And an outcome is for us, because we had to retrospectively measure apolipoproteins in this trial, can we predict treatment benefit in this pharma trial? And in order to see that you can maybe identify the patients which will have most benefit from additional treatment above statins with alirocumab. Okay, so how did we do that? Normally, you would ideally want to set up yourself a randomized control trial. So we had to use one which was already published, which was not tailor-made for our purpose. And normally you would have liked to have therapies which address your apolipoproteins. So we had to find out something else. So the alternative was retrospective measurement, as I mentioned, and looking with statistical modeling. And therefore we also needed the help of our statisticians to predict that treatment effect. So here we took the entire study populations at 2 time points, so nearly 12,000, and we measured at day 1 at baseline and then 4 months after treatment. And here you can see the number of incidents that we had. In the placebo group, I showed it already. First MACE was 12.2% and death 2.6%. In the treatment arm, we had reduction, 2.1%, and here only 0.3%. So this group was not very large enough even, although this was a huge effort to get all this analysis done. And the absolute risk reduction is shown here on the right-hand side. So it's just important that you know as a background that statins, they have a working mechanism in which they inhibit HMA-CoA reductase. So then you get less cholesterol in the cells, and then you get an upregulation so that the clearance is intensified and that you get lower LDL in the circulation. Whereas the PCSK9 inhibitors, they inhibit in the recycling and the internalization of the LDL receptor. And that's also a mechanism in order to enhance uptake of not only LDL, but also it's partially also helping to reduce LPA. These were the results that we found at the 2 time points for the entire study across all the apolipoproteins, the medians and the interquartile ranges in the 2 arms. The major effects were scored on apo-a because alirocumab is working on it, and also on apo-B, where you saw a more than halving of the median. And we had some extra effect on C2, C3, and E because probably they are keeping the lipoproteins in a soluble state in the circulation. So this is what I said. And then how did we do the statistics to prove any treatment benefit from doing this? Because that's our outcome parameter. So we also looked into logistic regression analysis with that cubic spline, and we constructed models. And in fact, 2 models have been constructed, one for the placebo group and one for the treatment group. And both models were then run on the entire study population, and we worked with probabilities for each participant. So what is the probability on an event using those 2 models if patients are allocated to the placebo to the placebo group as compared to the treatment group. And we calculated the treatment benefit from the subtraction of both probabilities. This is all statistically sound. It is, and as you can see here, if you look into the calculated risk if allocated to placebo as compared to treatment group for MACE and for cardiovascular death, then you can see that there are more points here. So that means that there is treatment benefit overall, because this is the line of identity. So more patients moved into this direction. And this is another way of the treatment effect. Of course, we want it to be, as we saw, an absolute risk reduction of 2.1%. So we tried to visualize this. And here you can see, for instance, on the right-hand side, a distribution of the probabilities based on that apolipoprotein profile, in this case for MACE, and if allocated to the placebo, you can see, depending on this treatment effect, that we can distinguish who has benefit on the purple here and who has no benefit. And that could help us if we use thresholds, but I can't step into that now, that we can help them guiding who has most benefit from doing this. So if we look then, again, translate that in areas under the curve, then you can see in this case, if for determining the treatment effect, lipid profiles have this area under the curve, which is quite good. Apolipoproteins can add, and the combination is not better. But here also we could, with this statistical approach in an existing randomized control trial, which is not the best design for us, because it was majorly focusing on cholesterol, which was already highly treated, we can still show an improved prediction of treatment benefit in this population. And here you can see it for cardiovascular death, what that means. Here it's opposite. The lipids, they have this area under the curve. Apolipoproteins can predict, but they are not better than the lipids in this case, and this is for the combination the same. So with this approach, we tried now for the first time, I think, that in an existing trial, SCT, in which outcome variables had been documented, that we tried to predict the estimated treatment benefit of that new medication on the endpoints in this population. And I think that is quite new. And all these data now can be generalized then. I think I told it already a few times, the study design, and because I had to shop, I had to look around who is willing to collaborate with us, because nobody is willing to pay such a lot. You can't set up from scratch such a randomized control trial. So we had to do it with what we had, a highly selected group with no specific attention to remnant disease and women, for instance, or only partially, intensively treated and already having a medication which is majorly meant for lowering LDL and not so much for the other apolipoproteins, except then apo-a. And but still, the positive news is that we could demonstrate clinical value of the profile. At least you saw at the level of clinical performance that we can predict from the baseline apolipoprotein profile the events, and also for treatment benefit. That's the clinical effectiveness side. We can identify in a better way. So therefore, the role as an add-on test would probably be feasible. Okay, but to determine its full potential, we are still busy with a similar study in obese patients, also that is done prospectively, and we hope that we will catch there more females and also more remnant disease. So to conclude, apolipoproteins, they should be measured as a profile, is our statement, because you saw the differences. There is no discrimination feasibility when we use them single, and they can improve, they have prognostic performance for events and cardiovascular death. And if we look into effectiveness, clinical effectiveness, you could see that they can improve prediction of estimated treatment benefit. And that means in that clinical care pathway, we hope that on the right-hand side here, that we indeed can add that this will hopefully motivate clinicians to look also into apolipoprotein profiling as an add-on test compared to serum lipids. It should, but we have to give more evidence also in the NEO study that we are still undergoing, improved characterization of dyslipidemia, and hopefully also better targeting of therapy, because these new therapies are very expensive, so that we can step into more biology-driven diagnosis and treatment of patients with further reduction of residual cardiovascular risk and a more personalized and effective approach. And I wanted to end with a few slides from the American Heart Association. There are very nice articles currently there, and also with a presidential advice telling us that, and that's nice, all our specialists are of course more or less organ specialists, and we should not only focus on the heart, but they are telling that we need a much more holistic approach. We are of course facing a pandemic of obesity, of diabetes type 2, and this is costing enormous amount of money. So, and all these people, if they have to be treated, we should try to keep them out of the hospital, to keep them longer healthy. So the clinical consequences of this tragedy is that we get now what we name CKM syndrome. So it's a combination of cardiovascular, kidney, metabolic. So the liver, the kidney, the heart, it's all affected by what the lifestyle and the way how we are living. And we should indeed, we are now on the return. So we have more mortality due to this. And we should, hopefully also with better diagnostics, we should reverse this, because diabetics, they have longer, they have a shorter median survival of 13 to 14 years, and the same for the very heavy people. So much has to be gained there. And I hope that the apolipoprotein profile or the ceramides or whatever, I think we will need a much more complete approach instead of focusing on single biomarkers in such complex diseases. And to end up this CKM syndrome, it has 5 stages, and now they start in fact with telling either you have no risk factors, or you are too obese, you have too much fat or dysfunctional adipose tissue. And this is then, I think this is the category where risk factors come along, and I hope that the apolipoprotein profile may, with time, if we can get this published, and after it's well criticized, that it will find its way. I would like, this is of course a joint effort. I talked about the Odyssey Outcome Steering Committee, so the statisticians which had to give us advice, that's the cardiologists in my department. Of course, many companies had to enable this. These are my research people, and this is the work of a PhD candidate, so she still has to give her, to get her PhD, but she did wonderful work by trying to do this. So I would like to thank you for your attention, and I'm happy to take questions if there is still time. Yeah. Any questions?
Sweat here. It's
If there are no further questions, my colleague and I would like to give a small wrap-up. So, because it's the end of this IFCC Scientific Division Satellite Symposium, it was the first time that the IFCC Scientific Division organized a collaborative meeting of this kind to inform the lab medicine community about mass spectrometry, an established technology that can overcome some of the challenges we face with current laboratory assays. The primary objective of this event was to inform the community about the strengths and challenges of this new technology by providing information on current applications in areas such as endocrinology, and to provide an outlook on potential future applications. As with all clinical lab activities, the quality of data produced is of utmost importance. Therefore, it was important to provide information also about opportunities and challenges related to quality with this technology. And it's so good to see that the people not only aim for new applications, but that there was also clear attention to develop guidance documents and other means to make this technology work on a daily basis. And we also see that mass spectrometry clearly evolves from a niche technology for very special applications to mainstream technology with broad applications and use. We learned that it has potential to go beyond what we currently are doing and provide new and more customized patient care. Still, it remains a technology that requires that requires a very high skill level, special lab infrastructure, and therefore it's not immediately affordable and feasible for many labs. Some may remember that we had a similar situation back when we introduced immunoassays, automated immunoassays, when they became popular. So it is the demand of the clinical lab community for better quality data that pushes innovation. And now, with the dawn of getting new, fully automated, closed mass spectrometry systems, we think we are getting closer for this technology to be more broadly used. We think we learned that mass spectrometry is not a solution to everything, and it will not replace technologies we have today in our labs immediately, nor will it be the only new technology entering the clinical lab. As a profession, we need to make sure current and future clinical and lab specialists have the appropriate knowledge and skills to use mass spectrometry in an appropriate and meaningful way, no matter if it's an open research type system or fully automated closed system. And with that, we hope this meeting, and we means Kostas Hubert, my colleague here, and myself as organizers from this part, we hope that this meeting was helpful, not only to those already familiar with mass spectrometry, but more importantly to those new to this technology, because it's an educational effort also. And I hope the information provided helps and will help you further to be prepared for these new opportunities that are ahead of us. And finally, we would like to thank the Greek Society of Clinical Chemistry and Clinical Biochemistry for their very kind support and collaboration, specifically Professor Tsatsanis and Mrs. Kurbasi for their great assistance in getting all the tasks done in a timely manner. And with this, we would like to close the session, and we thank you for being here. Thank you very much. Yeah.